everyone, my name is Cheryl Boggs and I'm going to be guiding you through this first part of our photosynthesis unit. Today what we'll be talking about is kind of an overview of the process of photosynthesis as well as delving a little deeper into the first phase of photosynthesis, which is known as the light-dependent reactions or sometimes simply the light reactions. So we're going to be talking about a lot of details about the process of photosynthesis, but really all of that is secondary to understanding the big picture of what goes on in photosynthesis. So those details help to support that big picture, but you need to make sure you understand this for sure. So this is just a very simple schematic of what's going on in photosynthesis. And so our definition that we'll be working from is photosynthesis is the process by which photoautotrophs convert the energy in sunlight into the energy stored in organic compounds. Now, hopefully you're familiar with the first law of thermodynamics. And the first law of thermodynamics says that energy can neither be created nor can be destroyed, but can be transferred or transformed from one form of energy into another. And so as we look at our reactants, which are the sunlight, the carbon dioxide, and the water, which we see again in this very simple diagram going into the leaf, and we see oxygen and glucose coming out, the sunlight is where the energy is coming into the system. So the sunlight is the input of energy. And that energy in sunlight is converted through the complex steps of photosynthesis into, as what's shown on the diagram, glucose or organic compounds. So let's kind of make sure we understand this definition. An autotroph literally means a self-feeder or an organism that can feed itself. And so those would be organisms that don't rely on consuming other things or other organisms to obtain their energy. And so algae, plants, some bacteria would be autotrophs. Anything able to do photosynthesis is an autotroph, as well as some organisms that are able to do chemosynthesis, which is not the focus of today's lesson. And then what is an organic compound? So we're talking about photoautotrophs converting the energy in sunlight into organic compounds. Well, glucose is an example of an organic compound, but an organic compound is one that contains both carbon and hydrogen. So methane, CH4, would be an organic compound. Sucrose, which is a disaccharide, would be an example of an organic compound, an amino acid, a protein, lipids. So all of those things that contain both carbon and hydrogen would be organic compounds. Something that does not contain one of those, it needs to contain both. So carbon dioxide, CO2, is not an organic compound because it does not contain hydrogen, even though it contains even though it contains carbon. So how does this fit into our big picture of ecology? So here we see our ecological pyramid, and hopefully you're familiar with the fact that the energy of the producers, again those autotrophs, form the base of the food pyramid, the ecological pyramid. So we need many producers to support a smaller number of primary consumers, since a lot of that energy that the producers capture is used for their own purposes, is lost as heat, is expelled as feces, and so not all of that energy is available to be transferred to the next kind of level of the to the next trophic level in the ecosystem. And so we need to have, in order for an ecosystem to be kind of self-sufficient, there need to be many more producers than primary, secondary, or tertiary consumers. And so this energy that photosynthesis, the process of photosynthesis, is able to capture and make useful is really important for not only the producers or those photoautotrophs who are able to actually obtain that energy, directly producing the glucose or the other organic compounds from using the sunlight, but also for everything else that follows them in the food web. Okay? And so that biomass that is generated by those producers really is important for supporting all of the living things at all of the trophic levels, not simply the producers. And here's just, again, this should be review for you, but autotrophs are responsible for capturing energy and fixing it, and we'll talk about what that means in the second module, into organic compounds. Okay? And heterotrophs like us, like fungi, like some bacteria, like some protists, consume compounds that are produced by other organisms to obtain their energy. So to be alive, means that one of the characteristics of being alive is that you consume energy to kind of power your processes. And so the difference between autotrophs and heterotrophs is just where they get that energy from. Autotrophs are able to kind of self-produce it, while heterotrophs need to rely on other organisms for their energy. So leaves are where most of the photosynthesis in plants takes place. And so certainly other living things other than plants do photosynthesis. But for our purposes, we're going to focus on plants. But as I mentioned earlier, algae and some bacteria are capable of photosynthesis as well. And so it's helpful sometimes to think of leaves as organs, which is what they are for plants, much in the same way that your kidney or your liver or your lungs is an organ in us, and in our bodies. And our organs serve specific purposes, and so do leaves. And so leaves are an example of the organ in plants that are specialized for photosynthesis. And so we've got multiple components here to the leaf, and it's important to understand kind of the different pieces. So here, this first, the kind of top layer is the cuticle, and the cuticle is a waxy layer, and its primary purpose is to help prevent the leaf from losing too much water through the process of transpiration. And so hopefully you remember that waxes are lipids, and lipids are hydrophobic because of those non-polar covalent bonds that hold the carbon and the hydrogen together in those long fatty acid tails. And so, just like you would if you wax a car and it rains, the water just kind of beads right off, right? This thick, or this waxy cuticle is helpful in kind of retaining the moisture inside of the leaf. So, kind of a thought question for you. In what sort of environment would you expect to see leaves that have a thicker cuticle than average? You would expect that in a drier environment, right, where transpiration would be more of a problem. So where the difference in water potential is much greater between the inside of the leaf and the atmosphere. Then we have the upper and the lower epidermis layers, and much like our skin, or the top layer of our skin, the epidermis, this is kind of just an outer barrier for the leaf. Then this, let's see here, this layer here that kind of goes all the way around, and it goes deep, through the plant as well. This is the palisade mesophyll. And mesophyll kind of means in the middle, and so hopefully you can sort of see that these cells here are kind of in the middle in terms of the cross-section of the leaf. These little green circles in here, okay, this diagram isn't perfect, but these are chloroplasts. Okay, so these are the organelles where photosynthesis takes place. And sometimes students have a hard time with scale, and so it's helpful to remember that in this picture what we're looking at is we're looking at a piece of one leaf, okay, and then all of these individual guys right here, these are cells, okay, and inside the cells we have the chloroplasts, okay, and inside the chloroplast is where kind of the magic happens of photosynthesis. Now, for simplicity, these cells have been drawn without the nuclei, without the mitochondria, without the endoplasmic reticulum. All those things are in there, but for our purposes right now, we're just focusing on the chloroplasts. Okay. Here we see it, as I just revealed, the spongy mesophyll. So hopefully you can see a little bit of a difference between the palisade mesophyll and the spongy mesophyll layers. And the key thing that should be jumping out at you is the spacing between the cells. And so the spongy mesophyll, the cells are much more spread out. There are more air spaces between the cells, and those air spaces are where the gases sort of reside. So the carbon dioxide that is taken in to be used during the Calvin cycle, as well as the oxygen that is produced prior to its being released from the leaf. And so kind of all of those spaces is where between the spongy mesophyll cells is where the gases hang out. Okay. Here we have the stoma. Stoma literally just means opening, and I haven't done this perfectly, but what the arrows should be pointing at really is the hole that is in between the two cells that are flanking it. So the stoma is actually the hole. And then on either side of each stoma, or plural stomata, we have what are called guard cells. And guard cells open and close to kind of regulate the passage of water as well as gases, carbon dioxide in and oxygen out, to facilitate photosynthesis, prevent a process called photorespiration, which we'll discuss, and also release water through the process of transpiration. Okay. Here we have, and it's the purple, the cells that are shown in purple on this diagram, those are the bundle sheath cells, and those will become important as we start talking about a type of adaptation that some plants possess in order to help them avoid a process called photorespiration. So the bundle sheath cells kind of go around the perimeter of what we call the vascular bundle here, so the xylem and phloem. So the xylem and phloem are the vascular tissue found in vascular plants, and they're responsible, the xylem is responsible for transporting water. Xylem is actually a dead tissue when it's functional, so it's got lignin in the sides, kind of in its cell walls there, and lignin is a polar molecule, and so is water. So hopefully you remember that about water. And so the structure of the xylem, though not the focus of today's lesson, it's still a good thing to review. The structure of the xylem, it's polar, and so is water, which is what it's transporting. And so the water not only sticks to itself through the process, or through cohesion, right, with water molecules being attracted to one another and forming hydrogen bonds, but it's also, the water is also attracted to the partial positive and partial negatives of the walls of the xylem, which kind of help it to creep up against gravity from where it comes in to the plant through the roots, and then all moves. Xylem is unidirectional, so the transport of water moves only from roots to shoots. It doesn't go in both directions. Okay, and then the phloem is responsible for producing sugars, and the phloem travels or moves sugars or organic compounds from what we call a sugar source to a sugar sink. So a sugar source would be an example of a leaf, right? Something that is producing sugar, and it transports that sugar to a sugar sink, so something that is either storing or using sugar. So that could be a root, right? Roots can't perform photosynthesis since they don't have access to light, but they can store it. Think of you know, a carrot or something like that. Or it could be transported to a part of the plant that's, you know, growing, or that's a flower that's not producing their own sugars. So the phloem travels kind of, or the contents of the phloem sap travels in both directions, from sugar source to sugar sink, while the water in the xylem travels only from roots to shoots. Now hopefully you've noticed that the palisade mesophyll and the spongy mesophyll are in red, and that is because that is where most of the photosynthesis is going to happen, is in those types of cells within a leaf. Okay? So if you've ever been outside and seen a leaf, hopefully you can see that leaves have a lot of surface area, and that's a huge advantage, and that is a theme that should be coming through to you loud and clear through your AP Biology course, is the more surface area that, you know, a structure has, kind of it's easier to get more materials kind of across that boundary. So think of something like the alveoli in your lungs, right? There are more of them that are smaller, so you're able to get improved gas exchange. Well, in much the same way, there's a lot of surface area in leaves, and that helps to facilitate more absorption of sunlight. And the more energy you have coming into the system, then the faster that photosynthesis can happen, and the more products that can be produced. And we'll discuss that a little bit. But are there any drawbacks to leaves having a large amount of surface area? Well, there are. And the more surface area that leaves have, yes, they can capture more sunlight, but they're also prone to more transpiration. And that can lead to desiccation, which is simply the drying out of the leaves. And so there are plants that live in areas or in conditions like deserts or other dry places where this is a real concern, where plants can lose way too much water to be able to survive. So through the process of evolution, there are some adaptations that certain species of plants have developed over many, many generations through differential reproductive success that have led to some adaptations that we'll discuss in Module 2. So now that we've talked about the structure of leaves, let's delve inside the cells of the leaves, moving into the palisade and spongy mesophyll. And again, I find that students often have a hard time with scale, and so hopefully this is, not dumbing this down too much, but in eukaryotes, right, organisms whose cells have a nucleus, photosynthesis takes place inside chloroplasts, which is what you see on your screen, which are inside cells, which are inside leaves. Okay, so there's multiple kind of levels of organization here to remember. So chloroplasts have three membranes, and again, membranes should be something that are kind of, coming up a lot in your AP biology study in terms of separating things into different compartments, and you'll see why that's important in just a few minutes. But we've got our outer membrane, as you can see on the diagram, the inner membrane, and the thylakoid membrane. And the thylakoid membrane in chloroplasts is folded many, many times into these tiny little discs that you can see here. I like to think of them as like little York peppermint patties all stacked on top of each other, and those are called thylakoids. So thylakoids, again, hopefully you're seeing there's a lot of surface area there, right? Rather than just kind of having one sort of large stack or one large kind of structure that didn't have all those foldings around the thylakoid, certainly we get a lot more surface area with this pattern of folding, which gives us our thylakoids. And then thylakoids are arranged in stacks that are called grana. Granum is the singular term. And then we've also got the stroma. And the stroma, on this picture, it looks just kind of like empty space that's surrounding the grana. But stroma is actually it's mostly made of water, but it's a fluid that is surrounding the grana inside of the chloroplast. And then chlorophyll, which is a pigment, and there are other pigments as well involved in photosynthesis. Those molecules are embedded inside the thylakoid membrane. So hopefully when you think of chlorophyll, you think of the color green, and we'll talk about why that is. But those molecules are embedded inside the thylakoid membrane, which is why the thylakoids appear green. So, little physics primer for you. But visible light is made of different colors of light with different wavelengths. So, right, we see white light typically. We don't see the red and the yellow and the green light. But think about a prism, okay? So hopefully you've all seen a prism before where a beam of light is shone into a prism, and then you can see basically what's at the bottom of the screen here. You see the full spectrum of visible light from the violet all the way to the red, okay, and everything in between. And so even though that's not the way that our eyes typically perceive light, that is actually what's going on. Okay? And so light has what we call a dual nature. It acts, kind of has properties of being both a wave as well as being a particle. And so that's a little bit of a tough thing to understand sometimes, but for our purposes today, we're going to mostly focus on the particle nature. And a particle of light is called a photon. Okay, so you can't say light is a wave and light without saying that light is also a particle or has kind of properties of both. So it's a little bit of a paradox, but that's how light operates. So a pigment is a molecule that absorbs light energy, and so we've already mentioned, right, that light energy or sunlight, usually most specifically, is what kind of where the energy enters the equation of photosynthesis. So if there was no light, there would be no ability to produce glucose or other organic compounds, since there's no energy coming into the system. If you don't put gas into your car, it's not going to run. So it's kind of the same idea as that. So pigments are molecules that absorb light energy, and the best way that I can sort of think of to describe this is pigments are kind of like light collecting buckets. Okay, so if it was raining $100 bills from the sky, you would probably run outside, right, and try to capture as much of that money as you could. And the best way to do that would be with lots and lots of buckets, imagining, of course, that the money just disintegrated when it hit the ground. So in much the same way, a pigment is like a bucket that helps a plant or another photosynthetic organism to absorb the energy and light. Okay? And so you have different pigments, or organisms do, that absorb light of different wavelengths. And so you can think about this again. You have multiple buckets to absorb light. So if you didn't have a bucket, 4 feet to your left, and money was falling there, you wouldn't be able to catch it. You wouldn't be able to kind of get it, imagining it was burning up when it hit the ground. And so, again, in much the same way, it's an advantage to plants to be able to, or other photosynthetic organisms, to be able to have multiple photosynthetic pigments, multiple buckets to capture light. Because if they were missing, if they don't have a pigment to capture a certain wavelength of light, that light would be not able to be absorbed, and so would not be able to be used for the process of photosynthesis. So we've got kind of a few major groups of photosynthetic pigments. Chlorophyll A is sort of our major one, and that's what you see here on the right hand side. That's the molecule of chlorophyll. Chlorophyll A, excuse me. We also have chlorophyll B, which differs slightly in structure. And then we have the carotenoids, and the carotenoids include xanthophyll and the carotenes. And hopefully you can kind of see the advantage of having multiple pigments. So an example would be, chlorophyll A absorbs wavelengths of light. Well, let's move on and we'll talk about this. So here, what you see right here is an absorption spectrum. Okay, so we see the different wavelengths of light with the different colors. So the shorter wavelengths in nanometers, so about 400 is kind of the violet end of the spectrum, and around 700 is the red end of the spectrum. And then we're seeing on the left-hand side the amount, kind of relative amount of light that is absorbed at each wavelength, and by which pigment, type of pigment. And so, which colors of light are absorbed well or not, and not well. And this is, something that you should be able to take a look at this and see that, red, blue, and violet light are absorbed really quite well by these pigments. But that yellow and green light, really, if you take a look down here, kind of between the maybe about 525 and 640 or so, there's not much light being absorbed at all. Okay, those lines are nearly flat. So taking a look at this, what would be the advantage to an organism of having multiple pigments? Well, the advantage is that with more buckets or more pigments, you can capture more light. And so, for example, between about, 670 or so and 690, this light right here in this range is primarily being absorbed by chlorophyll A. And so if chlorophyll A was not here, all of this light here would not really be absorbed, okay, because chlorophyll B isn't absorbing it very well, and from about here to here, from about 680 to 695 or so, there's really no absorption other than what's being done by chlorophyll A. And so again, if you don't have a bucket to capture that light or a pigment to capture it, you're unable to use it to drive photosynthesis. Okay. So again, taking a look at graphs are really key in terms of AP biology, and being able to take a minute and look at them and try to figure out the information that they are telling you. So up here on the top, we see an absorption spectrum, very similar to the one I just showed you. Okay, so what information does this tell you? Well, it's telling you the wavelengths of light that are best absorbed by each type of pigment. Okay, so the relative absorption of each wavelength of light, which corresponds to the color, okay, of light. And so again, as we just mentioned, the absorption spectrum is showing that the pigments are absorbing the red and the blue-violet light kind of the best, and they're reflecting the other colors. So that's why the plants are going to appear green to us, because they are reflecting that green light back to our eyes. They're absorbing the red and the blue and the violet, but the green is being kind of reflected back to our eyes. Okay. What information does this action spectrum show us or tell us? The action spectrum shows how the wavelength influences the rate of photosynthesis. And so certainly, again, key to reading any graph is to look at the labels on the axes because they're there for a reason. So what we're really seeing here, hopefully, is that there's some photosynthesis happening at almost every wavelength of light. So maybe with the exception of about 550 or so where it really troughs down there in the bottom, there's not much going on there. But again, these high peaks are really indicating that photosynthesis is happening at a much faster rate in the blue-violet light kind of range and the kind of orangey-red range, which corresponds to what we saw up top. All right, so the net overall equation for photosynthesis is 6 carbon dioxide plus 6 H2O, or water, in the presence of light yields C6H12O6 plus 6 O2. Okay. So, is photosynthesis an endergonic or an exergonic reaction? Well, if you've been paying attention and you know what those two terms mean, remember, an endergonic reaction is one in which energy is put into the system in order to make it happen. And an exergonic reaction is one where energy is released from a reaction. Well, in this case, photosynthesis is an endergonic reaction. Energy must be put into the system in the form of sunlight or light in order to make it happen. So as I mentioned at the beginning, photosynthesis occurs in 2 stages. The first, the light reactions or the light-dependent reactions, and the second, the Calvin cycle, sometimes called the Calvin-Benson cycle, or the dark reactions, or the light-independent reactions. Now, the light reactions or the light-dependent reactions require light directly, okay, and we're going to talk about exactly what they need light for in just a moment. The Calvin cycle, sometimes called the light-independent reaction, the Calvin cycle doesn't directly require light, but it does require the products of the light reactions. And so if you can follow that logic with me for just a second, if we don't have any light, the light reactions can't occur because they're light dependent. And since the Calvin cycle requires the products of the light reactions, or the light-dependent reactions, if they're not occurring, if the light reactions are not occurring, the Calvin cycle cannot occur either. Okay, so this is very kind of stepwise. Step 1 must come before step 2, and if step 1 is blocked, then step 2 cannot occur. Okay. So sometimes it's helpful for us to follow the energy in photosynthesis, okay, to kind of remember that photosynthesis isn't a process where we're really concerned about transforming energy. And so sometimes we can get lost in that, and the key thing here is to remember that first law of thermodynamics, that we can't create or destroy energy, but that it can be transformed from one form to another, which is what we're doing, or these photoautotrophs are doing in photosynthesis. So energy comes into the process of photosynthesis through light. Okay, it is transformed into ATP and NADPH, and that occurs through the light reactions. Okay, which is the first stage of photosynthesis, which occurs in the thylakoid. So those little green discs stacked one on top of each other, kind of like York peppermint patties. Stacked on top of one another. That's where the light reactions happen. That's where the light is captured and converted into energy in the form of ATP along with some NADPH. And then the Calvin cycle, which is the second step, uses that ATP and NADPH that was produced in the light reactions. Calvin cycle occurs in the stroma, so in that fluid that's surrounding the thylakoids, to produce organic compounds, which we commonly talk about as carbohydrates. Okay, so kind of the overall picture here is we're taking, or again, photoautotrophs are taking the energy and light which is not usable in terms of driving cellular processes and converting it into a form in which those processes can occur, or which can be used, that energy can be kind of harvested or used to drive cellular processes like active transport or other processes, muscle contraction, that require energy. So again, those organic compounds may or may not be used by that specific organism, but that is the whole point, is to convert the energy from an unusable form, light, into a usable form, organic compounds. So we're going to go ahead and skip this animation for right now, but it's a great thing for you to go through on your own to watch this animation as we start going through the photosynthesis process, the details. Okay? So, the light reactions of photosynthesis involve the use of what we call photosystems. And photosystems are a cluster of pigment molecules, and they're bound to proteins along with what's called a primary electron acceptor. And the basic role of a photosystem is to capture light, light energy. Okay, so these are our buckets. That's what we're talking about here. A whole bunch of chlorophyll or P700, chlorophyll or carotenoids, pigment molecules that are kind of all bound together to capture light energy. Okay. There are two different photosystems that are involved, and they were named in the order in which they were discovered. And so we have photosystem 2, which is also called P680, and it's called P680 because it absorbs light best at the wavelength of 680 nanometers. Okay. And photosystem 1 is called P700 because it absorbs light best at a wavelength of 700 nanometers. So I'm going to go through, I've broken this up into kind of an arbitrary set of 6 steps. And this can be a little bit confusing for people, but we're going to go through an animation that hopefully will make it clearer for you. So what we have here first is photosystem 2, which remember is a group of pigment molecules, P680, okay? And photosystem 2, which kind of is acting as an antenna, absorbs the energy in a photon, which is a particle of light, and as a result of absorbing the energy in a photon, an electron is excited away from photosystem 2 and it goes to a higher energy level. Okay? Now photosystem 2 is 1 electron short of what it requires. It's just lost an electron. It's been excited away to a higher energy level. Okay? So there needs to be a mechanism in place in order for photosynthesis to continue indefinitely to replace that electron. And that electron gets replaced through a process called photolysis. And hopefully if you look at that word, it makes sense to you. Photo means light, lysis, to split or to break or to burst. So photolysis is the splitting of water using light. Now, when photolysis occurs, one of the electrons is kind of delivered to photosystem 2 to replace that one that was lost. But kind of the byproduct, the side effect, so to speak, is that oxygen is released as a byproduct. Now, by splitting water, oxygen is released again. That's not the purpose of photosynthesis. That's not kind of the major point or what these organisms are, doing. But certainly for us, that's a very valuable kind of byproduct that's being produced. So if we think about this, which do you think came first? The process of photosynthesis or organisms that performed aerobic cellular respiration, that performed cellular respiration that required oxygen? Probably photosynthesis. And why? Well, it's hard to perform aerobic respiration when you don't have any oxygen, or at least not very much of it. So this is really key. And again, just kind of use your problem-solving skills to think through that. A process that produces oxygen almost by mistake would need to come before organisms that require that molecule in order to survive. So this is kind of just a generalized schematic of what we're talking about, and it's a very good diagram. I'm going to skip over it for now, but please feel free to use it if it's one that makes sense to you. Okay. So moving back to our kind of 6-step process here. So we had our electron that's gotten excited away from photosystem 2 when it was hit by a photon. We've had that electron replaced in photosystem 2 by splitting water through the process of photolysis, releasing oxygen as a byproduct. But now let's move back to our excited electron. The excited electron is going to travel down an electron transport chain. And an electron transport chain, kind of think of like a fire kind of bucket brigade, right? Old time, they used to put out fires, right? Somebody's down by the river and then they fill up their bucket and they pass it to the next person. It's kind of like that, okay? But instead of individuals holding buckets being what makes up the electron transport chain, in the electron transport chain what we actually have are a series of cytochromes, and cytochromes are proteins. And as the electron transport chain continues, each cytochrome is progressively more electronegative than the cytochrome that came before it. And what it means to be electronegative, remember, is it's an atom's ability to attract electrons to itself. So basically each cytochrome is more attractive to the electron than the previous one. So the electron keeps moving from one electron that it thought it liked to one that it actually likes even better, and then one that it likes more than that, so to speak. Okay, so it's attracted, which is why it continues to kind of move down the electron transport chain. Now, as it does that, it's losing some of that energy that it gained when it got excited by being hit by the photon. Now, again, that first law of thermodynamics says you can't lose energy, right? And so what we have is a really awesome example of something called a coupled reaction, or coupled reactions, where one process is sort of losing energy, so to speak, but that energy is being used to power another process, okay? So how that happens is as that electron loses, again, so to speak, energy as it moves down the electron transport chain, that energy is actually used to build up a concentration gradient of protons. And so what we're trying to do, or what is happening here essentially, is pumping protons from one side of the membrane to another in order to build up an area of high concentration and have an area of low concentration, right? So when you have an area of high concentration next to an area of low concentration, that's a concentration gradient, and that represents potential energy, the separation there of those kind of differing areas of concentration. And that's a process that we're going to use essentially, or is going to be used to perform something called chemiosmosis. Okay. Now, at the same time as all of those first three steps are happening, the same thing is essentially happening to photosystem 1. So photosystem 1 is, a little bit physically separated from photosystem 2, still within the thylakoid membrane though. Again, it's another group of pigment molecules, and it absorbs light energy. Okay, one of its electrons it gets excited to a higher energy level as a result of being hit by the photon. Okay, so again, the key thing kind of here for step 3 is we had our exergonic reaction where our electron was losing energy as it moved down the electron transport chain, but that's coupled to an endergonic reaction, one that requires energy, the pumping of protons against their concentration gradient. Okay. Now, same situation in photosystem 1 as what happened in photosystem 2, right? One of its electrons got excited away, and so it's one electron short. So this electron in photosystem 2 is actually replaced in a different way than the splitting of water. It is replaced by the electron that was excited away from photosystem 2. So ultimately, the source of electrons is water, right? Because that's what provides the electron to replace in photosystem 2. But the electron in photosystem 1 is replaced by the electron that we previously discussed, which was excited away from photosystem 2. Now, in much the same way as what occurred to the first electron, the second electron, the one that's excited away from photosystem 1, also travels down an electron transport chain, which is made of increasingly electronegative cytochromes. Again, losing energy, so to speak, as it goes. Being able to power, again, the movement of protons against the gradient to build up a concentration gradient. And ultimately, that electron reduces or is added essentially to NADP+. And when that electron and its corresponding hydrogen are added, we get a molecule called NADPH. And NADPH is an electron carrier. Okay, so what you need to basically think about for NADPH is an electron babysitter. It's going to shuttle electrons from one place to another. It's an electron taxi is maybe a good way to think about it. Okay, so let's talk about this. We talked about the fact that those electrons are losing energy as they move down the electron transport chain. Okay, and the energy that those electrons lose, so to speak, is used through the process of chemiosmosis to make ATP. So what's all this fuss about ATP? What is this molecule? Okay. Well, here it is. Okay. There are some pieces to ATP. Okay. So what we have is adenine. Okay. Adenine is a nitrogenous base. Where else do we see adenine? ATP, ATP, of course, DNA and RNA, right? It's part of the nucleotide there. We have ribose, which is a 5-carbon sugar, okay, a carbohydrate. And we have 3 phosphate groups, and that's kind of where the name of ATP comes from. It's adenosine triphosphate. Okay? Now, that's wonderful, and ATP is used, right? Hopefully when you kind of hear the word ATP, you think, oh, cellular energy. Okay, this is where this is what cells use for energy, and you'd be correct, right? But you need to understand why that's the case. Okay? The reason why ATP is such an excellent molecule to be used for cellular energy is because it's very unstable. And you might not think that it's very unstable, but take a closer look, and it's the phosphate group tail that's really what's very unstable. And the thing that makes ATP such an unstable molecule are these negative charges. Okay? So you see these negative charges that are all right next to each other. Well, what do we know about negative charges? They repel, right? So these phosphate groups really that are all stuck next to each other are very unhappy to be so near to one another in terms of chemistry because they have like charges. It's kind of like, forcing 3 siblings who can't stand each other to sit in the back seat together for a long car ride, okay? One is trying to get out as quickly as possible. And so basically what happens is it's very easy to break the bond here and pop this 3rd phosphate group off. Okay, and if this 3rd phosphate group wasn't here, what we'd be left with was ADP, right? Adenosine diphosphate. And so that's how ATP is used to do cellular work. So when this phosphate group is essentially popped off, okay, that phosphate group, which is removed, is added to another molecule. And that's a process called phosphorylation, right? Adding a phosphate group to something. It's phosphorylated. Usually when that happens, it activates whatever molecule the phosphate group is being added to. So if the phosphate group is added to a protein, it typically activates it, or an enzyme, which obviously is a type of protein as well. Okay, that's usually done by a type of enzyme which is called a kinase. So kinases are responsible for phosphorylating other molecules and activating or turning them on. Okay, so ATP is an excellent molecule for cellular energy use because it's very unstable. It can be that kind of final bond there between the 2nd and 3rd phosphate groups can easily be broken to perform cellular work through phosphorylation. So what's going on here? So we have protons. Protons are hydrogen ions, okay, and they are pumped from the stroma. Okay, remember that fluid we talked about around the thylakoids into the thylakoid space, and that happens across the thylakoid membrane. Okay, so we had, remember the kind of the wrapper of the York peppermint patty can be our represent our thylakoid membrane, and the inside of that York peppermint patty would be the thylakoid space, so the interior lumen of the thylakoid membrane. Now, I said they're pumped. What does that imply? That should make you think, oh, active transport, right? Pumping implies energy is needed to do it, okay? We're not talking about diffusing here. We're not talking about osmosis. We're talking about pumping, pushing something where it doesn't want to go, essentially, right? Based on the concentration gradient. So where do we get the energy from to do this? For pumping, we need energy. Well, this energy came from, remember that electron that was, those electrons that were losing energy, right? That exergonic reaction as they were traveling down the electron transport chain. That exergonic reaction was coupled to this endergonic reaction. And so the energy that the electron lost moving down the electron transport chain was used to pump the protons across the thylakoid membrane. That's going to build up for us a concentration of hydrogen ions in the thylakoid space. And I'm going to show you a picture of this in just a moment. Okay? And that concentration gradient is going to represent potential energy. Where you have a difference in concentrations on either side of the membranes, you have potential energy. Now, in the process of chemiosmosis, okay, and chemiosmosis is the movement of ions across a selectively permeable membrane down their electrochemical gradient. So it's an ion, right? A molecule with a charge, and it is moving across a selectively permeable membrane like the thylakoid membrane down their electrochemical gradient. So the protons are going to diffuse back to the stroma. Remember, we built up this high concentration of them in the thylakoid space. They're going to diffuse back to the stroma, and we're going to let them do that, but they're going to move through a molecule called ATP synthase. And this is called the proton motive force. So the protons are moving, right? That's just where they get their energy from. And as they kind of move through the molecule of the ATP synthase, you can think of it kind of like a water wheel or one of those kind of revolving doors that they have at some office buildings or things. So ATP synthase spins as these protons are kind of flowing through this molecule to get to the other side, to the area of lower concentration to achieve equilibrium. ATP synthase is going to spin kind of like a turbine, and it's going to jam ADP and the phosphate group together. Well, why don't they want to be together? Why does it require so much force? Well, remember, ATP has 3 negative phosphate groups all next to each other. They're not happy to be there, so it's going to take a lot of energy to force that 3rd phosphate group onto a molecule of adenosine diphosphate. But when we do that, or when that's done, we have a molecule of ATP. Okay? So let's move through this animation. Hopefully this will be helpful to you. So what we see here is kind of inside this green area here, this is our thylakoid membrane, okay, our York peppermint patty wrapper. Out here we have the stroma, and down here we have the thylakoid space. So here we have our photosystems, photosystem 2, also known as P680, as well as photosystem 1, which is P700. Okay, so we've got all of our chlorophyll in here, which is responsible for absorbing the energy in light. Now, just for kind of illustrative purposes, I've said that each of these photosystems has 5,000 electrons. That's not accurate. I have no idea how many electrons are in each photosystem. That's not important. But what you need to understand, or the reason why I put this here, is you need to know that there's a finite limit to the number of the electrons that each photosystem has. So, those little orange guys that I just put up, each of those individually are cytochromes, okay? And those together, kind of the first 3 there on the left is 1 electron transport chain, and the other one on the right is the 2nd electron transport chain, okay? At the end of the 2nd electron transport chain is NADP+. So this is a molecule that is waiting for another electron and its corresponding hydrogen to form NADPH. So it's kind of waiting. Down here at the far end we have ATP synthase, okay, that protein that we talked about that spans the membrane. It goes on both sides of the membrane, and ACE, ATP synthase, right, it's an enzyme. Here we have a whole bunch of protons, hydrogen ions, and our hydrogen ions are at the moment, as you can see, equally distributed on either side of the thylakoid membrane. So as of right now, there is no concentration gradient, right? They're at equilibrium. There's no desire for, based on kind of molecular movement, for any of these protons to cross the membrane. Now that doesn't mean that one from the stroma might not just diffuse, into the thylakoid space, and one from the thylakoid space might not diffuse into the stroma. Remember, at equilibrium there's still movement of molecules, but there will be no net movement of molecules, kind of, unless there's some sort of outside kind of influence, which we're about to get. Okay. Also out here in the stroma we have our ADP molecule and our phosphate. Now, if we could put them together, what would we get? ATP, you're exactly right, but since they don't like to be near each other, it's going to take some force, and at the moment we don't have the force to do that, so stay tuned. Okay. Down here also in the thylakoid space we have our water molecule, okay, our little Mickey Mouse guy, and he's just going to hang out for right now. Remember that water is a reactant required for photosynthesis to occur, and hopefully you'll see why in just a moment. Alright, so let's start this. Here we go. We have light hitting the photosystem. Okay, so sunlight, that's what those little yellow guys represent. Okay, and when the light hits the photosystem, there we have an electron, okay, and an electron from each photosystem gets excited to a higher energy level. Okay, now what happens as a result? Well, if you notice, we had 5,000 electrons when we started. this whole process. But now, since each photosystem has lost an electron, each photosystem is now down to 4,999 electrons. Well, we're fine for now, but if we keep going and going and going and don't replace that electron, eventually there will be no electrons left and photosynthesis would have to stop. And so there needs to be a built-in way, if this process is to kind of keep happening, to replace those electrons that get excited away, and we'll talk about what that is. Okay? So following our first electron, okay, I'm going to do that one more time. So over here on the left-hand side, we had our electron that was excited away from photosystem 2, and it's going to move down the electron transport chain through those cytochromes. And remember, that's an exergonic process, so energy is being lost. But that is coupled to an endergonic reaction, right? Remember that we talked about these protons being at equilibrium. So they're not on their own going to, a whole bunch of them diffuse from one side to the other. They're already at equilibrium. But watch what happens as our electron loses energy as it moves down the electron transport chain. Okay? It's coupled to the movement of protons. And so we're starting to build up a concentration of protons here inside the thylakoid space. We already have a higher concentration now of protons in the thylakoid space than we do out in the stroma. Okay? Now that electron ended up at photosystem 1. So instead of 4,999 electrons, now photosystem 1 is back to 5,000 electrons. So back to good, okay? That photosystem has replaced its lost electron, no problem, ready to go again. Okay? Now let's follow the second electron, which was the one that was excited away from photosystem 1, P700, and was excited to a higher energy level. So it's going to move down its electron transport chain, okay? Again, in much the same way, it's going to cause, kind of through coupled reactions, cause the movement of the protons from the stroma to the thylakoid space, and then that electron and its corresponding hydrogen are going to reduce NADP+ to form NADPH. There we go. And NADPH, if you'll remember, is like an electron taxi. So it's just going to hold on to that electron for now, and we're going to use it a little bit later on in the Calvin cycle. So hopefully you can see, and hopefully you would agree with me, that right now we have a large concentration of protons or hydrogen ions here inside the thylakoid space, and a very low concentration of protons or or hydrogen ions in the stroma. But before we talk about those, let's talk about photosystem 2, because we haven't had, that hasn't been kind of rectified yet. Oh, no, I lied. Okay, so we're going to talk about the protons first. And hopefully what you can see here is that high concentration of protons down here in the thylakoid space, low concentration in the stroma. Well, just by, because of this electrochemical gradient, these protons want to move essentially from the thylakoid space to the stroma, and the way to do it for them is to go through ATP synthase. It's a win-win. Okay, so they are moving back to where kind of they want to be in terms of electrochemistry, but as they move through ATP synthase, think of it again like a revolving door, and it's going to jam the ADP and the phosphate group together, producing ATP. Okay, so they're moving down their concentration gradient. This is why it's chemiosmosis, right? But it's producing ATP for us in the process. And this would continue. I just did one of those animations, but this would continue right until the concentration gradient was eliminated and both sides were at equilibrium. So now let's go back to photosystem 2, P680, where we were talking about the fact that they are still down one electron. Okay, and again, hopefully you can see the implications of that if the electron was not replaced prior to continuing with photosynthesis. So here's where our handy dandy water comes in. Okay, so we have light hitting the water. Okay, this is the process I mentioned earlier called photolysis, the splitting of water using light. Okay, and when that happens, what we end up with is an oxygen, okay, and 2 hydrogens. Now here's what happens. The electron from one of those hydrogens returns to goes to photosystem 2, and now we're at 5,000 electrons. Okay, we're back to good. So the splitting of water needs to occur to replace this electron that was lost, right, when this one got excited away. So it doesn't need to be the same one. It certainly isn't going to be the same one, but we need the same kind of overall number of electrons. But what that leaves is some hydrogens and hydrogen ions and some oxygen. And the hydrogen ions are used to kind of help build up this concentration gradient here in the thylakoid space. And the oxygen, once this happens, again, this is happening very quickly, once we would get another oxygen through the same process, right, by splitting another water, those oxygen molecules would join together, producing O2, and that oxygen would be released through the stomata out into the atmosphere. Okay, so this is where we get the oxygen from in photosynthesis. Here's another animation that's essentially showing you the same thing, showing the light hitting the photosystem, some electron movement, the protons being transferred from one side of the thylakoid membrane to the other. The other thing that should be mentioned is the pH is going to be different as well. So inside the thylakoid, as you get more and more protons, remember that protons are acidic. And so if you're building up a higher concentration of protons inside the thylakoid, you're going to have a more acidic environment in the thylakoid space than you are out in the stroma. This is another animation that you can take a look at. You just need to click on it, and that's something that you can certainly do at your leisure. Good way to review what's going on. So there are multiple, this is a hard concept sometimes to understand if you just kind of read the bullet points, but you want to understand kind of where electrons are moving. So hopefully one of these multiple animations is something that will help you. So as a quick recap of what we talked about with the light reactions, in the light reactions, the energy in light is used to excite electrons, and those excited electrons are used to make ATP through the process of chemiosmosis and produce NADPH, which is an electron carrier, which the ATP and the NADPH are used to power the Calvin cycle, okay, which is the second stage of photosynthesis. The light reactions occur in or across the thylakoid membrane, which is inside the chloroplast, which is inside the cells. Okay, so remember all those the two photosystems there that were embedded inside of the thylakoid membrane, and then everything that's going on either side. Light and water are required. Okay, so we call those the reactants. The light is required in order to excite the electrons in the first place. The water is required to replace the electron that is lost in photosystem 2. Okay. Oxygen, ATP, and NADPH are the products of the light reactions. Oxygen is produced through photolysis, right, when water is split in order to replace the electron that was lost from photosystem 2, and ATP is produced as a result of chemiosmosis, and NADPH as a result of the movement of the electron through the second electron transport chain. Now there's also something called cyclic electron flow. And cyclic electron flow is, or cyclic phosphorylation. Remember I talked about phosphorylation being the addition of phosphate to something, of a phosphate group to something else. In this case, cyclic phosphorylation is adding a phosphate group to ADP. Okay, and if we add a phosphate group to ADP, we are going to get, you guessed it, ATP. Okay. Now cyclic phosphorylation is a more ancient biochemical pathway, and how could we know that? Well, yes, some of us are much older than you are, but the key thing is, in terms of kind of general, I don't know, sort of rules to live by, the simpler a mechanism is, the older it is in terms of kind of evolutionary time. Okay, so, right, so to give you an analogy, right, the first vehicle that was built didn't have GPS navigation, right? If you put a 2013 version of a car next to a 1920 version of a car, you'd probably be able to tell the difference in terms of kind of the advances. And so cyclic phosphorylation is a simpler way, a simpler kind of mechanism of photosynthesis. It's also used by most bacteria that perform photosynthesis. And so since bacteria we know to be older, again, they're simpler in structure, right? They're prokaryotic, don't have a nucleus, and have other kind of simpler mechanisms just in general. Since they use it, and since all photosynthetic eukaryotes use it, we can be pretty confident that it's something that developed a long, long time ago in terms of kind of phylogeny, right? So as kind of different traits, as different organisms sort of split at various times in their history based on different kind of environmental conditions and mutations that were acquired. The key difference to know here is that cyclic electron flow produces ATP in much the same way as we just discussed with non-cyclic electron flow, but it does not produce any NADPH, okay? And this is actually quite helpful. It may not seem so at first, but the Calvin cycle does require ATP and NADPH, but it requires more ATP than it does NADPH. And so performing cyclic electron flow sometimes helps to make up the difference. So it produces extra ATP to make up for kind of the excessive or the greater demand for ATP that the Calvin cycle has over NADPH. A couple other key differences here. Only photosystem 1 is used, so photosystem 2 is not used. Electrons are what we call recycle, okay, which is why it's called cyclic electron flow. And water is not split, so photolysis does not occur. So here we have kind of a diagram of cyclic electron flow. Okay, I'm going to go through this diagram again through this little animation, but this time we're only going to show what happens during cyclic electron flow. So I'm going to set it up much the same way. Here are our photosystems. Again, there are a number of electrons. Our electron transport chains, NADP+, ATP synthase, our protons or hydrogen ions. Okay, so what happens here is light is going to hit photosystem 1, okay, and it excites its electron to a higher energy level, again reducing the number of electrons that photosystem 1 has from 5,000 to 4,999. Now remember the coupling of the exergonic and the endergonic reaction. So as this excited electron moves down the electron transport chain, which is exergonic, some of those protons are going to be pumped from the stroma into the thylakoid space, which is an endergonic reaction, okay? But now, instead of that electron being added to NADP+ to form NADPH, as it occurs in non-cyclic electron flow, in this case, the electron, we'll go there in a second. Okay, so this is much the same as what we talked about, right? That proton gradient, high concentration down here in the thylakoid space. So some of those protons are driven by the electrochemical gradient. This is called the proton motive force, right? These protons are moving. They're going to cause ATP synthase to spin like a turbine, jam that ADP and phosphate group together to make ATP. Okay, but here's where the key difference happens: is that electron now is cycled back to photosystem 1. Okay. And now photosystem 1 is back to its 5,000 electrons. Okay, so that same electron just kind of makes the cycle. Okay, it's excited away, moves down the electron transport chain, and then comes back. And so you might think, well, what's the point? Well, the point of this is that as that electron gets excited by light and then loses that energy, right, through the exergonic process of moving down the electron transport chain, it can be used to pump protons in that endergonic reaction from the stroma to the thylakoid space, which can be used to produce ATP. So let's compare these two. In non-cyclic electron flow, which is what we went through first, 2 photosystems are used, both photosystem 2 and photosystem 1. In cyclic electron flow, however, only 1 photosystem is used, and that's photosystem 1. Okay. In non-cyclic electron flow, water is split through that process of photolysis to replace the lost electron from photosystem 2. But in cyclic electron flow, water is not split. Photolysis does not occur to replace electrons. Instead, that electron that was excited away is recycled back to the photosystem, okay, reused. When non-cyclic electron flow occurs, oxygen is released, but when cyclic electron flow occurs, oxygen is not released, and hopefully that makes sense, right? The oxygen that comes from photosynthesis comes when the water is split. Well, if the water is not being split because we don't need to replace the electron, then oxygen is not going to be released in cyclic electron flow. In non-cyclic electron flow, NADPH is produced from using that electron from photosystem 1, but in cyclic electron flow, that electron is not available to produce NADPH because it is cycled back to photosystem 1, and so NADPH is not produced. In non-cyclic electron flow, ATP is produced. It is also produced in cyclic electron flow. Okay, so at the bottom of your screen here is another animation that you can use to take a look at to hopefully clarify the difference between cyclic and non-cyclic photophosphorylation. Okay. So to put all this together, what have we talked about today? So we've discussed the light reactions, or the light-dependent reactions. And what occurs in the light reactions is that the energy in sunlight is transferred into chemical energy in the form of ATP and NADPH, okay, to power the Calvin cycle, which we'll talk about in the second module. Light and water are required for the light reactions to occur. Okay, those are the reactants. ATP, NADPH, and oxygen gas are produced through the light reactions. Those are the products of the light reactions. Now, 3 of these things have asterisks next to them: water, NADPH, and oxygen gas. Any idea why? Well, that means the asterisks denote that those are not produced during cyclic phosphorylation, okay, as we just talked about. The other key thing that's not on this slide, but you really want to make sure you understand, is that light is certainly required, right? Well, actually, that was mentioned, I'm sorry. Chlorophyll is required in order to perform the light reactions, right? We need a light-absorbing molecule in order to capture the energy in sunlight. If you don't have a bucket to catch the light, you're unable to use it to kind of excite those electrons away. And the other thing that was not mentioned on here is you need intact thylakoid membrane. So that membrane that separates the stroma from the thylakoid space must be intact. If there's a break in that space, okay, if there's a break in that if there's a break in that membrane, then there's no way to separate into 2 areas, an area of high concentration and low concentration, because protons would then just kind of diffuse wherever they like in order to achieve equilibrium. And so if that was the case, they wouldn't, go through ATP synthase, and ATP would not be produced. So your thylakoids must be intact. So here we have a few a few animations that can show you again photophosphorylation and
Every chloroplast in a plant cell is packed with stacks of flattened sacs called thylakoids. The thylakoid membranes contain chlorophyll, as well as most of the other components required for the light reactions of photosynthesis. The chlorophyll-containing structures within the membranes are called photosystems 1 and 2. In addition to the chlorophyll molecules and other pigments, these complexes contain proteins. The photosystems function to absorb light energy and donate excited electrons to carriers in an electron transport chain. In an electron transport chain, electrons are passed from one electron carrier to another in a series of redox reactions. In the thylakoid membrane, the chain consists of carriers called plastoquinone, cytochrome, plastocyanin, ferredoxin, and NADP reductase. In addition to the electron transport chain, thylakoid membranes contain a type of protein complex called ATP synthase. ATP synthase uses the high concentration of hydrogen ions inside the thylakoid as a source of energy. The synthase taps this energy to create ATP molecules, which fuel later stages of photosynthesis. When a photon strikes a photosystem, its energy is captured by one of the many pigment molecules in the photosystem's large antenna complex. After absorbing the energy, the pigment molecule passes the energy to other pigments until the energy reaches a chlorophyll molecule in the antenna's reaction center. After absorbing energy, the chlorophyll molecule boosts one of its electrons to a higher energy level. The electron is held very loosely and easily jumps from the chlorophyll to an electron acceptor molecule. After losing an electron, the chlorophyll molecule is considered oxidized and carries a positive charge. The electron lost from the chlorophyll molecule must be replenished. In photosystem 2, water replenishes chlorophyll's lost electrons. After donating 2 electrons, a water molecule dissociates into 2 hydrogen ions and half of an oxygen gas molecule. The oxygen later enters the atmosphere. Meanwhile, the electrons continue along the electron transport chain. As these excited electrons reduce plastoquinone, energy is released and used to move hydrogen ions across the membrane into the thylakoid interior. As hydrogen ions build up inside, they become a source of energy that is later used to make ATP. Electrons move from plastoquinone, which becomes oxidized, to cytochrome, which becomes reduced. The redox reactions continue as cytochrome passes electrons to plastocyanin. From plastocyanin, the electrons will replenish electrons that are lost from a chlorophyll molecule in photosystem 1. When photons strike photosystem 1, the light energy is absorbed and transferred to a chlorophyll molecule in the reaction center. With each photon, the chlorophyll molecule donates an excited electron to an electron acceptor. Electrons from plastocyanin replenish the electrons lost from the chlorophyll molecule. Excited electrons from photosystem 1 are used to reduce ferredoxin. NADP reductase then uses 2 electrons from ferredoxin and 2 protons from the stroma to reduce NADP+, producing NADPH and hydrogen. NADPH is later used to fuel the energy-consuming sugar production stage of photosynthesis. During these light reactions, hydrogen ions accumulate in the thylakoid interior. These ions will have a tendency to diffuse to regions of lower hydrogen ion concentration outside the thylakoid. The hydrogen ions move through ATP synthase, which uses the flow of hydrogen ions to power the production of ATP. ATP fuels later stages of photosynthesis.
This will trace the pathway of carbon dioxide through the process of photosynthesis, which is a little more applicable to the second module since we haven't discussed carbon dioxide yet.
Photosynthetic reactions take place in the chloroplast and can be divided into 2 pathways. Within the thylakoid membrane, the energy transduction pathway converts light energy into ATP and NADPH. The second pathway takes place in the stroma and uses energy produced by the first pathway to incorporate carbon dioxide into carbohydrates in a process called carbon fixation. The sequence of reactions by which carbon dioxide is converted into carbohydrates was uncovered in the 1940s and 1950s by Melvin Calvin, Andrew Benson, and colleagues at UC Berkeley, and this pathway now bears their name. How did these scientists determine the pathway of carbon dioxide fixation in photosynthesis? Like other researchers, Calvin and Benson were interested in the basic photosynthetic process of using light energy to convert carbon dioxide and water into carbohydrates and oxygen. However, there was no obvious way to follow the path taken by the carbon contained in carbon dioxide as it became incorporated into other molecules. In the mid-1940s, however, nuclear reactors were being built, and scientists could use these reactors to create radioactive isotopes of common atoms like carbon. In nature, carbon exists as the carbon-12 isotope, but a radioactive isotope of carbon, carbon-14, could now be produced that behaved almost identically to carbon-12. A useful tool was thus available for tracing the path of carbon dioxide in photosynthesis. Calvin and Benson first constructed an apparatus consisting of a light source and a flask that could be filled with photosynthetic organisms. For this purpose, the researchers chose the unicellular green algae chlorella. To label metabolites, the researchers injected carbon-14 labeled CO2 into the flask and then quickly killed the algae and extracted their metabolites in boiling ethanol. Using a technique called two-dimensional paper chromatography, the researchers were able to separate the components they sought. In this technique, the plant extract is first spotted in one corner of a piece of filter paper. The paper is then suspended in a chamber containing a solvent in such a way that the solvent just touches the edge of the paper. Capillary action will pull the solvent across the paper. As it passes over the extract, it will pick up various components. Based on their molecular properties, the components will travel at different speeds as they're pulled along the piece of paper by the solvent. In order to improve the separation even more, a second run was performed in a perpendicular direction. By placing an X-ray film on top of the paper, the locations of the radioactivity labeled compounds could be determined. When the chlorella was exposed to carbon-14 labeled CO2 for 30 seconds, a variety of compounds were found to be labeled, including organic compounds, amino acids, and sugars. All of these compounds had incorporated carbon from the 30-second exposure to carbon-14 labeled CO2. But the authors wanted to determine the first compounds to become labeled, so they continued to reduce the reaction time until only a single compound dominated the chromatogram. This compound, which became labeled after just a few seconds of exposure, was identified as a 3-carbon sugar phosphate called 3-phosphoglycerate, or 3PG. The researchers eventually identified the acceptor molecule for carbon dioxide as the 5-carbon compound ribulose bisphosphate, or RUBP. Catalyzed by the abundant enzyme ribulose bisphosphate carboxylase oxygenase, or Rubisco, the carbon dioxide becomes incorporated into the RUBP molecule, which is then converted into 2 molecules of 3-phosphoglycerate. Using a combination of chromatographic and chemical techniques, Calvin and his colleagues eventually identified all of the intermediates within the cycle, and Melvin Calvin was awarded the Nobel Prize for his work in 1961.
Where does the oxygen come from? Well, hopefully you have a good idea about that by this point in the module, but this is again another animation you can watch to talk about the source of oxygen and really understand where it comes from.
In plants, water enters through the roots, while atmospheric carbon dioxide enters through openings in the leaves called stomata. Oxygen and water are released by the plant through these same openings. Photosynthesis takes place within the leaves in specialized organelles called chloroplasts. The basic process of photosynthesis can be summarized using the equation shown here. Carbon dioxide, water, and light energy combine to yield the products of sugar, oxygen, and water. Of course, knowing the reactants and products does not tell us the specific nature of the photosynthetic reactions that take place. For example, where does the oxygen that's released into the atmosphere by plants come from? Does it come from the carbon dioxide or from the water? This question was answered in the 1940s by Samuel Rubin and colleagues at UC Berkeley using isotopes of oxygen. The common isotope of oxygen is oxygen-16. By using the oxygen-18 heavy isotope of oxygen to label either the carbon dioxide or water reactants, the source of the oxygen released into the atmosphere can be determined. Let's begin by labeling the carbon dioxide reactant with the oxygen-18 isotope. Our data will consist of the O18-O16 isotope ratio measured in the evolved oxygen as compared to the starting ratio in the CO2 reactant and unlabeled H2O. Note that not all of the CO2 will contain the heavy isotope. Now let's repeat the experiment by labeling the H2O reactant. Again, our data will consist of the O18-O16 isotope ratio measured in the evolved oxygen as compared to the starting ratio in the H2O reactant. What can we conclude from this experiment? Is the source of oxygen released by the plants derived from the carbon dioxide or water reactants? When CO2 was labeled with 18O, none of the oxygen released was labeled. However, when O18 labeled water was used as a reactant, the oxygen released was labeled with a heavy isotope. Therefore, the source of the oxygen in the O2 released by the plant is water.
And then this final animation, I guess, from this source shows a couple of experiments that were used to demonstrate how chemiosmosis occurs, and I encourage you especially to watch this one at your leisure. This is an online animation, so you'll need access to the internet to access this. And it's an animation of the entire process of photosynthesis. So it might be a little bit premature, the second part, in order to, because we haven't discussed the Calvin cycle yet, but it's a great animation overview of the entire process of photosynthesis.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
That brings us to the end of Module 1. I hope to see you in Module 2. Good luck!